comes into question, these two components can then mix. And this protoxin then mixes with the activating enzyme, thus cleaving the glycoside and then having a localized and often concentrated delivery of toxin to the insect. So the focus of the talk will actually be focusing on perhaps one of the most famous, maybe cyanogenic glycosides can take this title as the most famous, but glucosinolates, at least a very close second, activated defense. And the reason they're so famous is because they are present in a lot of our food crops, specifically the Brassicaceae mustards. And maybe what gives that mustard its nice potency and wasabi is actually from a glucosinolate. But importantly, these compounds are also present within the model plant Arabidopsis thaliana. And chemically, these compounds are N-hydroxysulfate esters with a beta-thiolinked glycoside. And they are usually divided into various categories based on the amino acid in which they're derived from. Aliphatics being from valine, leucine, methionine, benzenic coming from phenylalanine, tyrosine, and indolics coming from tryptophan. The main compound that I'm going to be kind of focusing on today is actually the aliphatic glucosinolate, which is actually the main glucosinolate within Arabidopsis thaliana, which is phloem SOB glucosinolate. And in true two-component defense fashion, this compound is separated from its activating enzyme known as myrosinase. And once it cleaves this glycoside, it forms this unstable agglucone. And this agglucone can rearrange to various products, nitriles, thiocyanates, depending on the chemical environment around it, and also maybe some specifier proteins that might be present. But what's thought to be the most important toxin within this mix are these potent electrophiles known as isothiocyanates. So already this is kind of, I think it's quite incredible, this type of organization in this defense, but this can become even more organized in Arabidopsis, where you actually have dedicated cells to deliver even more concentrated blasts of this defense. These so-called S cells or sulfur-rich cells containing high levels of glucosinolates, as these are very sulfur-rich compounds, and then dedicated idioblasts in close proximity containing high concentrations of myrosinase. And it's important to note for this talk that actually it seems that this type of defense is very well localized surrounding important tissues such as the phloem. Okay, so that's our chemical component of this part of the talk. Now I want to introduce you to the insect side. Now these compounds, these activated defenses, are usually associated with chewing herbivores. Why is that? Well, that's because chewing herbivores, at least superficially, and of course it's actually true, they elicit the most superficial damage that we can see through the mastication of this plant tissue. This causes the greatest amount of mixing of these two components, and thus it's thought that these defenses work particularly well for these insects. But less is known about piercing, sucking herbivores, and the ones that feed on phloem and xylem, because maybe their tissue damage is a little bit lower, and thought that these compounds don't really pertain to these insects. And that's really what some of the work of my dissertation focused on, and what I'm presenting to you here today. So now I want to introduce the insect that I'm going to be talking about throughout the entire rest of my presentation today. Is the silverleaf whitefly, Bemisia tabaci, which is a phloem feeding insect. And the reason that I was studying this insect is severalfold. First of all, I was part of a project called the African Cassava Whitefly Project, where we were investigating ways to control these extremely large outbreaks on cassava in sub-Saharan Africa, but also the fact that Bemisia tabaci is an extremely polyphagous insect. It feeds on over maybe 1,000 plants characterized to date. It's also, not only is it prolific, but it's also quite damaging in several ways. So what is a phloem feeding insect? Well, of course, as their name implies, they feed on the phloem, and they do this through utilization of a uniquely adapted stylet that they use to navigate between plant cells, sampling cells along the way so that they know where they are until they get to their sugary rich food source. And of course, because they're not destroying all of the tissue that's surrounding it and mixing it up in their mouths, it's kind of it goes without saying that this is usually kind of a lower tissue damage type of feeding. But they can still do a lot of damage. For example, once they get to the phloem, they feed continuously, and the frass or the poop of this insect is known as honeydew, and they kind of deposit this on the leaf as they feed. And this is actually, I took a photo of a cassava leaf in sub-Saharan Africa that's actually cracking from the amount of mold, because this honeydew is a perfect substrate for the growth of various sooty molds. So as you can imagine, this drastically reduces photosynthetic potential of these plants. To add insult to injury, it just so happens that like other sucking insects and piercing insects, they also happen to be perhaps the most efficient vectors for various plant diseases, which can basically make, especially for crop plants, the tubers in cassava, for example, necrotic, and that you can no longer basically use these as food sources. Okay, so besides having a stylet and feeding, what other barriers are there as a phloem feeder to actually being one? And these are usually two, but I'm going to add a third one today. And these are most often the nitrogen barrier and the sugar barrier. Now, with regards to the nitrogen barrier, this kind of pertains to just the concentration of amino acids, this juxtaposition, the replete nature of the nitrogen content within the phloem in comparison to the extreme amounts of carbon that you can see in the forms of sugar that are within the phloem. And phloem feeders utilize symbiotic bacteria actually to overcome this as they produce the amino acids for them. The second is the sugar barrier. And this actually pertains to just the extreme concentrations of sugar that's present within their food source. And when you feed on such high sugar-containing substrates, you're likely to dehydrate and die. And so these insects must be really good at osmoregulating. And one of the ways that they do this in conjunction with another method, but one of the main ways that they do this is turning lots of different osmolytes of sucrose, for example, into long-chain sugars, basically making the honeydew that they excrete isotonic to the gut tissue of the insect itself. Okay, so those are those two barriers out of the way. And then, of course, the one I focused on was, are there also allelochemical barriers, specifically two-component defenses? Do these also represent an actual defense to these insects? So this is kind of like how I want to set up the progression of this talk is kind of question leading towards these to the research. Is that, do these phloem feeders actually have contact with these activated chemical defenses, and do they actually constitute a defense to them? And then the next subsequent question is, have they developed any mechanisms to detoxify them if they are defenses? Maybe any unique mechanisms to detoxify? And then finally, what are the enzymes that are doing these reactions if they are actually doing something? So with regards to the first one, do two-component defenses, and we're focusing now in on the glucosinolates from Arabidopsis thaliana and other Brassicaceae, do they actually constitute a chemical defense towards phloem feeders? And a very simple kind of experiment to look at this and as a proxy for understanding the diet of the insect, because pure phloem is actually extremely difficult to get, we decided to look at their poop. We decided to look at the honeydew of these insects, because what goes into them and what comes out is maybe a good idea of what is actually happening within the insect. And I want to return to this activation pathway of the glucosinolates, where you have your intact glucosinolate interacting with myrosinase, and you form these various toxic compounds, the most potent of which is the isothiocyanate. And what I didn't show you before was that there exists kind of a very ubiquitous method for detoxifying electrophiles, and that is the mercapturic acid pathway, which involves the action of glutathione S transferases. And basically what glutathione is a tripeptide, and it's responsible for dealing with these electrophiles, which would otherwise perhaps interact with proteins, making them non-viable. And there are also downstream scavenging reaction mechanisms to kind of mitigate the loss of nitrogen in this conjugation. And when we looked at the honeydew, after concentrating it down, what we were able to see, lo and behold, is that indeed we were able to show 4-MSOB isothiocyanate, the nitrile, but most importantly, we were able to detect 4-MSOB glutathione and downstream scavenging reaction N-acetylcysteine. And this was important because this showed us that activation was taking place within the insect as it was feeding, and it was being detoxified via an old mercaptoric acid or a ubiquitous mercaptoric acid pathway. So these compounds could actually represent toxins to these low damage phloem feeding insects. Okay, so since we have a possible defense compound, what are the methods that these insects use to detoxify them? I already talked about one being the mercaptoric acid pathway, but that's after activation. What other methods are there to detoxify? Previously in literature, it has actually been shown that glucosinolates could be desulfated, and this actually makes it that the activating enzyme can no longer activate them. But nevertheless, we wanted to look deeper into the honeydew, see if we could find anything else. And when we did, in an untargeted approach, look into the honeydew, we saw, importantly, intact 4-MSOB being excreted. But close to that, in clustering, there were these features of very similar isotope pattern and fragmentation pattern to 4-MSOB glucosinolate. But they had, when we looked a little bit deeper into them, we noticed that they were increasing in mass and having earlier and earlier elution times in a reverse phase HPLC system. And if you can do the math really quickly in your head, you'll notice that as from each mass difference, you have a mass difference of 162 mass units, mass overcharge units. And this, if you're not readily familiar with conjugation detoxification chemistry, this is usually or almost always associated with the addition of a hexose, and this is usually a glucose moiety. So we went to work on purifying these compounds from the honeydew utilizing HPLC fractionation and a method and column that can actually separate these compounds. It doesn't look as pretty as I'm showing it here, but a method that could separate these guys. And then also we decided to analyze them via NMR so we could get their structures. And this kind of workflow that I'm showing you here is really simplistic in its nature, but this part of it is really important and extremely difficult because the amount of honeydew that we can actually get from these insects is extremely small. Every single one of these insects is about maybe a millimeter in size. And therefore, the amount of honeydew that they produce is extremely small. But thanks to the wonderful NMR facilities that we have here at the Institute, and perhaps even more of the wonderful NMR operators, we were able to get structures of micrograms of amounts of material from the honeydew, which is really fantastic, because getting a lot more than that would have been very difficult. And lo and behold, what we were able to show was that indeed these insects were glucosylating these glucosinolates, 4-MSOB glucosinolates, sorry, on the beta-thio-linked glycoside. And this beta is very important because the linkage mode was actually alpha-1,6, which is different from the original plant beta-linked glycoside. And for one glucose addition, we were able to show from the purified compounds, you have either an alpha-1,6 configuration or an alpha-1,4. And then for the diglycosylated compound, you have a serial alpha-1,6 addition. And these are the compounds that we've actually managed to purify. But in an untargeted approach, we were also able to see signals for up to 3 additions and up to 4 additions. Okay, so these insects were taking 4-MSOB glucosinolate and they were glycosylating them. And in comparison to all of the other detoxification strategies that I mentioned, this one is actually the vast majority. This represents the vast majority of all the 4-MSOB that goes in is converted to these glycosides. Okay, so is this actually a detoxification reaction? And in order for something to be a detoxification reaction, it would have to kind of fulfill this criteria that when it's the 4-MSOB glucosinolate is glycosylated to any n degree, it can no longer serve as a substrate for the activating enzyme, and therefore it can no longer produce all of these downstream toxic products. So a very simple experiment kind of showed this, where we took honeydew, which, as you recall, it was very important to us that it contained this intact form SOB glucosinolate as well. And we just, in control reactions, incubated with water and then with the purified myrosinase. And we could quite plainly see that the parent form SOB glucosinolate is readily degraded while the insect compounds persist. So in conjunction with that first discovery of the activation being present within these insects, this now most likely represents a potential detoxification reaction. Okay. Oh, and that's exactly what I said there. So what about other glucosinolates that are present within Arabidopsis? Because form SOB is the majority, but there are other ones present. And we were actually able to see that it can do this, that Bemisia tabaci, feeding on various glucosinolate-containing plants other than Arabidopsis as well, can glycosylate them to varying degrees depending on the side chain. And now one that I want to draw your attention to is the indolic glucosinolates. Previously in literature, it's been shown that indolic glucosinolates represent a potent anti-feedant activity towards aphids. And interestingly, the Bemisia tabaci whitefly was actually not so good at producing glycosides of this indolic and actually preferred to form a majority of a desulfated form of this metabolite, which is also inactive to myrosinase. However, I also want to add, interestingly, when we looked at a glucosinolate specialist whitefly, Alerodes protella, that feeds on Brassicaceae almost exclusively, it actually doesn't desulfate these compounds anymore. It only glycosylates them, and it's very good at it. So that was really nice to see. Besides glucosinolates, can this be extended to other types of compounds? So we just kind of did artificial diet feedings, and I'm going to explain to you in a bit how we do this, at least the methodology of it. And we were able to show that form SOB glucosinolate, of course, is capable of being glycosylated by these whiteflies. The related cyanogenic glycoside linamarin is, but there does seem to be some specificity, as in there's a cutoff depending on the side chain. So the variability also is important. And of course, this is a very large question. Is this actually a unique kind of reaction? Because of course this is the first kind of description of these serial glycosylated metabolites. Were we just missing them before, or and now that we actually know what we're looking for, we can detect them in all of these other insect frasses and maybe other honeydews. But when we did look at insects that were feeding on plants containing these glucosinolates, form SOB glucosinolate, for example, we were actually able to show that only phloem feeding insects seem to produce these compounds, and none of the other feeding guilds were actually producing them, which shows that this is kind of a phloem feeding guild specific process, which is pretty interesting. Okay, and now the last part, question 3, I hope you can see that. I don't know if you can because of the screen sharing, but I wouldn't be allowed to be in the Gershons and biochemistry department unless I ask the question, what are the enzymes that actually perform these chemical transformations? And when you think of phase 2 detoxification, glucosylation, there are several examples that come to mind. The reglycosylation of Dimboa with an inversion of stereochemistry, this very unique N-glucosylation of midazolam, and the seeming polyglucosylation of gossypol. And what all of these reactions have in common is the enzymes that carry them out, being UDPGTs, which are uridine 5-diphosphoglycosyl-transferases. And what these enzymes are known as Lelwar transferases, which largely kind of break down into 2 categories, being the inverting or retaining Lelwar transferases. And what makes them these types of transferases, they utilize a activated form of glucose in order to transfer onto the acceptor molecule in the form of a nucleotide diphosphate. Now, what that means is that these insects actually are investing energy in order to detoxify these. So we became very interested in investigating the mechanism of these glucose additions to these compounds. So in artificial diets, and now I'm kind of going to explain how we do that. Basically, we have a tube of insects where we sedate them in the cold so that they're kind of lying on their backs. And then we quickly pull a piece of parafilm over top of this tube, very thin, kind of to represent the surface of an abaxial leaf surface. And then we put a solution, which is the artificial diet, between another piece of parafilm on top, and then allow these insects to feed for 48 hours, collect their honeydew, and analyze it. And that's these artificial diet feedings. And what was present within these artificial diets was sucrose, because it has to be there. These insects need sucrose in order to survive and feed properly, pretty much, for any extended period of time. And also, of course, our acceptor molecule, our phloem glucosinolate. But the thing that was variable that we added were various sugars, being the fully carbon-13 labeled glucose and fully carbon-13 labeled fructose. And if we were expecting kind of a Lalois transferase, then we would expect to see label incorporation in the honeydew in the production of these products. But actually, when we looked at the honeydew of these products, and I'm showing you the MS spectrum of these compounds, we weren't able to see any type of label incorporation. So it doesn't seem that that is necessarily the case for what we're looking for, a Lalois transferase. There are other enzymes that can actually perform these reactions, though, and in a very similar set of experiments, and this is kind of one of my favorite figures because I think it's rare that an experiment can work so well the very first time you do it. I was extremely lucky with this experiment, but containing similarly sucrose and phloem glucosinolate, but this time the various labeled sugars are isotopomers of sucrose, fully labeled sucrose, glucose labeled sucrose, and fructose labeled sucrose. And when we looked at the honeydew and the products in the honeydew of those that fed on these artificial diets, we were able to see that beautiful label incorporation for +1 glucose corresponding to +6 mass units, +2 glucose, +6, +12, and then finally for +3, +6, +12, and +18. And this was perfectly mimicked in the glucose-labeled sucrose diets. But this was inherently absent, this MS pattern, in the fructose-labeled sucrose diets. And this is completely consistent with enzymes known as sucrase-transglucosidases. So what are they? If we return to that original slide that I talked about of those interesting enzymes that are responsible for osmoregulation, let's look at the mechanism. And I kind of say this in our lab meetings and everything ad nauseum, so I'm sure everyone who is attending from our lab is sick and tired of hearing this. But if you were going to design an enzyme that is responsible for osmoregulation, if you're going to design an enzyme for osmoregulatory purposes, this would undoubtedly be it, because it has a concentration-dependent fate. So after hydrolysis of sucrose, and you kind of have this liberation of fructose, fructose is taken up for energy purposes for the insect, you form this glucosyl enzyme intermediate, and this concentration-dependent fate is dependent on the amount of sucrose that's around. When you don't need to osmoregulate, water can enter the catalytic pocket more often, and you get overall hydrolysis to glucose, which is fine because you don't need to osmoregulate. But when you do need to osmoregulate and there's more sugar around, well, sugar may enter the catalytic pocket and you can get transglucosidation. And this can happen again and again and again. And you basically end up forming these, am I not longer in the presentation? Yeah. And you basically end up forming these long chain sugars that we were talking about. This is very reminiscent of what we were seeing with this detoxification reaction. Okay, so what are the potential enzymes in Bemisia tabaci that might be performing this? Well, it turns out that they're actually part of a family called the glycoside hydrolase family 13, specifically subfamily 17, or at least the best candidates would be from this family. And in order to, this is actually work from our collaborators in Israel, in order to actually narrow down the potential candidates which could do this reaction, because as I mentioned, this was the first time that this was being described. So knowing exactly what candidates we could look at was a little bit of a shot in the dark. But through expression analysis within this insect, as well as looking at the different clades we chose 5 different candidates to express. And I kind of just also want to draw your attention, because everything highlighted in red in this circular cladogram is actually from Bemisia tabaci. And we can see the extreme number of GH13 enzymes within this insect, even in comparison to other phloem feeders. And it also just so happens that Bemisia tabaci is perhaps one of the most polyphagous insects on this list, as even, I mean, especially in comparison to the 2 other phloem feeders represented there. So we had these 5 genes as potential candidates to express. And that's exactly what I did. And I'm showing here that, oh yes, I expressed these genes for production of protein in S2 insect cells. But in reality, what I did was I tried multiple different systems, and then the best system that worked, perhaps because it mimics the most native folding environment, was in insect cells. And in simple enzyme assays, I incubated both phloem-SOB glucosinolate as well as sucrose, and let these enzymes do their stuff. And I was able to show 2 different enzymes which were capable of transglucosidating phloem-SOB glucosinolate using only sucrose as a substrate. That's all that was added in the alpha-1,4 fashion. Unfortunately, I couldn't form the alpha-1,6, and we still don't know why, but we were able to at least show transglucosidation from these enzymes. And for the interest of maybe the biochemists out there, they were utilizing the exact same mechanisms as when I incubated with glucose-labeled sucrose. Only the labeled product was formed. Okay. Now, in order to actually understand if these enzymes were maybe really responsible for this, and perhaps we can focus more on Bt sucrase 5 because it behaved a little bit better in the in kinetic assays. What I did was basically determine KMs in saturating concentrations of each of the substrates, sucrose and phloem-SOB glucosinolate. And I was actually able to produce kind of physiologically realistic values for these enzymes, being around 200 millimolar for sucrose as a KM, and around 0.5 millimolar or 500 micromolar for phloem-SOB glucosinolate. And this is actually exactly what we would expect to be present within the phloem. So this was also very exciting. Okay, and the last thing that I want to touch on is just that I talked about osmoregulation as kind of like the primary driver for where this function maybe comes from. And you might be thinking, as well as we did, that maybe this is just an offshoot of osmoregulation, and then this isn't actually detoxification in any way. So I want to draw attention to the various concentration-dependent fates, but also the reaction with other sugars. And when we looked at these enzymes, focusing in on sucrase 2 and sucrase 5, I was actually able to show that sucrase 2 and sucrase 5 were the worst hydrolytic enzymes. And even though that they could produce glycosides of phloem-SOB, they did not, over control cells or any other enzyme that was present in these assays, produce glycosides of sugars themselves. So there seems to be this division between enzymes which modify sugars and enzymes which modify plant defenses. And to kind of add to this, I've now characterized enzymes that do indeed form transglucosidation products of sugars, but they do not touch secondary metabolites or any that I've tested thus far. So that was really nice to see as well, this division. Okay, and as a summary, I just want to quickly summarize that I was able to show through these kind of question-led study that 2-component defenses, despite them being kind of low damage insects, they do actually constitute a defense to phloem feeders. And phloem feeders detoxify these potential toxins via serial glucosylation. And this is a kind of unique detoxification to the phloem feeding guild. And they utilize this through kind of enzymes that were derived from primary functions such as osmoregulation. And with regards to future directions from this project, one of which I'm focusing on kind of now in my research while I'm here, before I leave the Max Planck Institute, there are many. For example, because we're working in Arabidopsis, we have kind of a nice utility to look at plants that perhaps contain glucosinolates and those that don't, in order to see the fitness effects that can happen with silencing. The large expansion of these GH13 genes in these insects is also interesting, and perhaps this is necessary when you want to increase your host breadth in phloem feeders. And finally, I'm also very interested in understanding what types of mutations or changes within the protein sequence are important for functional changes from an enzyme that can modify sugars to an enzyme that focuses primarily on secondary metabolites. And with that, I want to finish my talk. Hopefully I wasn't too long. And with thanking Jonathan and Daniel from my department, my supervisors for their guidance, the technicians, as well as my collaborators in Israel, Michel Reichelt, Christian Petz from NMR, Shai Morin, Oznat Malka, our lab assistants and secretaries, people listed here, our funding bodies, and all of you for your attention and your attendance in these talks. So thank you very much. I'm very looking much forward to questions.
Yeah, thank you very much, Michael. Great talk. And
York, okay, it was York. I looked at York's question thing in the chat, and I was I thought it was like a thing from you to say like, hey, you're talking way too long or something like that.
No, it was great. Yeah, if you have questions, feel free raise your hand or just put them in the chat and we can read them. Oh yeah, then we have the first question. Please go ahead.
Yeah, hi. That was a great talk. I'm curious, have you looked at the expression of the enzymes you found? Are these intracellularly localized within, I would guess, the midgut, or are they secreted? Do you know anything about where they're expressed in their localization?
Yeah, that's a great question. So, I mean, that was part of that was part and parcel for the selection of these enzymes for expression, because as I said, it was kind of a shot in the dark, but we also used expression of these genes within the insect gut, as well as their kind of sequence alignment with other insect transglucosidases, which have shown like kind of very small transglucosidase activity. So they are expressed in the midgut or in the gut of this insect. And with regards to further localization, at least in aphids, sucrase-transglucosidases have been shown to be localized further to these perimicrovillar membranes. And they're associated with these perimicrovillar membranes, which kind of anchored in, maybe not specifically anchored, it's kind of a little bit unclear, but they're sitting in between these guys and they're associated directly with the membranes, kind of sticking out into the gut lumen. And yeah. That's as much as I know. And yeah, these guys are more expressed. So we had like whole body versus gut transcripts from one study, and we used that in order to kind of narrow our targets down.
Do we have more questions? Otherwise, oh yeah, Gaurav.
Hey, Michael. So very nice talk, and I might have missed this. The enzyme assays that you did were based on enzymes that were purified from the S19 insect cells. Is that true?
So these were actually the active fractions that I utilized. I tried purifying these guys from insect cells. But when I did, I had no activity of the enzyme afterwards. So what I used, and that's why I couldn't do more detailed kinetics. I could only get the KMs of these enzymes because I utilized normalized by protein amount total, but I could only get these KMs by utilizing the kind of the media of these insect cells. So they're actually secreting these proteins as they're expressing them at the S2 insect cells for some reason. This has changed now because I figured out how to express these guys in bacteria, or rather I didn't figure it out. A wonderful Japanese group figured out how to express insect GH13s in bacteria properly just by adding some divalent cations that help perhaps activity and folding. And now I am actually able to properly purify GH13s from bacteria, which is nice. I should be able to get better KMs from these now. I mean, better kinetic data rather. Yeah.
Is it possible? So you show me all of this evidence is quite convincing, but is it possible that the physiological activity of the enzymes is different from what you see in these in vitro assays in the cells itself when they encounter?
It's a good question because, well, I mean, first of all, I think
I'm asking because they're part of this large enzyme family and they have promiscuous activities.
So first of all, these enzymes are known, at least oftentimes, to associate in dimers, and this can perhaps help in their transglucosidation capabilities in forming these long chain sugars in vivo. And I didn't investigate whether mine were in fact forming dimers or something like this by doing like cross-linking or anything like that. But for example, I could never form the alpha-1,6 for glucosinolates. I was able to form small amounts of the alpha-1,6 when I was looking at cyanogenic glycosides and linamarin, but never for glucosinolates. So the activity that I'm seeing of these enzymes is somehow different than what I'm seeing physiologically. But at least I guess we're in maybe the right ballpark of the enzyme family that we were looking for and maybe potentials. I definitely can't say definitively that this is the main enzyme that has the activity in vivo or something like that at this point. Thanks for the question.
Yeah, I also have a question. I think you might have, I think you touched it a little bit with this answer and the answer to the question before. But you showed very nicely that like this one whitefly species had like basically 71 copies of this enzyme family or these enzymes, and you selected 3. And I was like kind of wondering, or maybe I missed it, like how you selected those 3. And of course, I mean, it could be like any of the others that might have this activity. I was wondering.
Yeah. So this, that goes along with a little bit of the question before on there's 71 GH13 genes, which seem to be full-length coding genes that even have variable transcription depending on what plant this insect is feeding on. How we went about choosing them, it was kind of down from, this was done by our collaborators. We asked them to look at the expression profiles within the gut versus the whole body transcript. That was kind of the first thing we expected them to be within the gut more than anything else, although of course we can't narrow down that these aren't expressed perhaps maybe in the mouth and salivary glands or something like this. And then we also considered these honeybee transglucosidases that had kind of this alpha-1,6 transglucosidation capability. They saw some minor activity in these hydrolases for transglucosidation. So they also said, okay, which guys have the highest score in comparison to those guys, those genes. And then also it was kind of a, let's try and hit at least one from each kind of these major clades that seem to be separating. And then, that was the major criteria that we went on. It was somewhat of a shot in the dark to see what we could get, but some guidance based off of those other parameters that I mentioned.
Okay, just a very quick follow-up question. Is it possible that those are induced basically based on like feeding on the glucosinolate containing SAP, that they're kind of just being expressed afterwards?
Yeah, it's a good question. So there has been actually a significant amount of work done to look at just overall transcriptomes of these insects feeding on various plants. And there seems to be some variability between the species of whiteflies for inducibility. So it seems that Bemisia tabaci, or Miam1 Bemisia tabaci, is seems to be kind of more constitutive in its expression for almost all genes. It has very little variability across different plants. There is some, but it's not like, oh, all the other ones are turned off and all of a sudden this guy goes on, or I mean, even like something close to resembling that. It seems that it always has a lot of different genes that are turned on at all times, in comparison to feeding on maybe like even control plants. So we had ones that fed on eggplant as a kind of control plant, which is kind of like a universal kind of rearing for these whiteflies. Then you put them onto a cyanogenic plant like cassava, and then, or you have them on glucosinolate, Arabidopsis. Chili pepper or something was one of them that was also used. And it seems that like they always had almost the same genes that were always turned on. In some cases, we had some interesting genes that were turned on, but they were actually P450s or something like that. Yeah.
Okay, thank you. That's very interesting.
Thanks.
Do we have any more questions from the audience? Don't see anything in the chat. Don't see any raised hands. Okay, then I think, yeah, I want to thank everybody for being here today. And if you want to stay on for a little bit more time and chat a little bit, please feel free. And otherwise, we see each other again on April 1st.
Oh, yeah, thank you very much.